Hi class, in this recording we are going to be talking about nucleic acids, the final macromolecule of the human body that we haven't gotten to yet. So let's dive into it. As we think of nucleic acids, they are a polymer or a large molecule made up of nucleotides. So we have nucleotides as the monomer. If we take many nucleotides and combine them together to make a polymer of nucleotides, we call it a nucleic acid. There are two nucleic acids that we're going to focus on primarily in our bodies. First, we have DNA. DNA stands for deoxyribonucleic. Excuse me, my bad. DNA stands for deoxyribonucleic acid. Sorry about that brain hiccup right there. And then we think, as we look at RNA, RNA stands for ribonucleic acid. So as we look at DNA versus RNA, those are the two broad categories of nucleic acids that we're going to talk about. DNA tends to be the larger of the two. It contains millions upon millions. One molecule of DNA will have millions of nucleotides, and it's used to make our genes, and those genes contain the instructions for synthesizing protein molecules. As we look at ribonucleic acid, RNA, there are three kinds that we are going to focus on in this class. If you take a molecular or cell biology, you learn about more than three kinds of RNA. So as we look at RNA, the three kinds we focus on in this class are the three that we need for protein synthesis. So messenger RNA, mRNA, ribosomal or rRNA, and transfer or tRNA. As we look at RNA molecules, they tend to be much smaller, 70 to 10,000 nucleotides long, and they're going to carry out specific bits of information that we use for synthesizing proteins and then assemble the amino acids together to make the protein based on the information that was stored in the DNA. As we look at these two, the RNA versus DNA, RNA notably is single-stranded. It has one strand to it, whereas DNA is going to be a double-stranded helix. In our body, there occasionally will be double-stranded DNA, double-stranded RNA, and occasionally will be single-stranded DNA. But again, this is not molecular biology, so we're just going to say RNA is single-stranded, DNA is double-stranded. Now, within our nucleotides, or our nucleic acids, DNA and RNA, we have four base units. These four base units are cytosine, guanine, and adenine. And those are present in both DNA and RNA. So we have cytosine, guanine, and adenine present in both. However, in RNA, we have uracil, and in DNA, we have thymine, T. And this is something that a lot of students get mixed up between the two. Uracil is RNA only, thymine is DNA only. And as we're looking at these base pairs, or these nitrogenous bases that make up the nucleic acids, there are some base pairing rules. Cytosines will always pair with guanines. So you can see blue and green here are always going to be paired together in the DNA molecule. And then A and T are always going to be paired together. Yellow and red are always paired together within the DNA molecule as well to help give it its double-stranded nature. And then within RNA, A will base pair with U because U is a substitute for the T. As we look at an individual nucleotide, it is going to have three components. We're first going to have our nitrogenous base. In this case, our nitrogenous base is adenine. And then we're also going to have a sugar. In this case, our sugar is ribose. And then we're going to have some phosphate groups. In this case, we have a triphosphate, three phosphates attached. And this particular molecule that we have up here is an adenosine triphosphate. We have an adenine, nitrogenous base, plus a ribose. That's called adenosine when you combine those two together. And then because we have 1, 2, 3 phosphate groups, we call it adenosine triphosphate, or ATP. ATP is important because this is the energy currency of the cell. And as we're looking at ATP, this molecule is how we transfer chemical potential energy from a glucose to the specific chemical reaction that we need it in. And in particular, it's this third phosphate group right here that's highly unstable and very prone to being knocked off the ATP. And when we knock off that third phosphate group, that's when we release the chemical energy stored within the ATP. As we look at this ATP, we use it again for energy transfer. So we take energy that we gain from the exergonic reactions. When we think of exergonic reactions, these are reactions that are going to give off energy. And then it will release that energy for physiological work, whatever we need to do in our body, maybe moving a molecule against this concentration gradient, or perhaps we need to build a larger, more complicated molecule. So it's that second and third phosphate group that has the highest energy covalent bond. And as we cleave that particular covalent bond, also referred to as a phosphodiester bond, we are going to release a lot of energy. And that energy is going to be used for many different things within our bodies. We'll talk about many examples over the next 2 semesters. That's all we have for this recording. If you have any questions or comments, please feel free to post them on the class discussion board or shoot me an email. And as always, happy studies.